All right, welcome back. So in the digestion lesson, you saw how food polymers were broken down to their monomers in the stomach and small intestines. This was so the molecules were small enough to be transported by the blood and moved into the cells. We did that on the first day using these cartoon versions of the monomers and polymers, so the hexagons, triangles, circles, squares, rectangles. And then yesterday we did digestion with the actual atoms and molecules. So here's a protein being broken down to its component amino acids. Look at how water molecules are required to break apart that molecule. This is called hydrolysis. So hydro for water and lysis to break, hydrolysis. Here's starch being broken down to glucose, again, using hydrolysis or those water molecules. So now we have 4 glucose molecules. And here's fat being broken down to glycerol and fatty acids. Notice that the number of atoms in all of these stayed the same before and after digestion, and that the energy remained as chemical energy throughout this chemical change. The next step is for these monomers to be used for growth in a process called biosynthesis. Bio means life, and synthesis means to make. So when you're growing, you're essentially making life, biosynthesis. In this activity, we will be looking at what biosynthesis is and what is happening at the atomic molecular scale. Digestion takes place in the stomach and the small intestine. That's where our marker stayed for the majority of our digestion lesson. Now those food molecules are going to move on. The molecules are going to move into the blood and circulatory system where the heart pumps them to wherever they are needed. So go ahead and move your marker to the heart to show it in the circulatory system. And then in this activity, we are going to move these molecules from the bloodstream into a muscle cell in the leg of a cow. Move your marker to the leg of the cow. In this, let's review what's in that leg muscle. Cow muscles are essentially beef. So here's a nutrition facts label for beef. As you can see, beef is composed primarily of fat and protein. Muscle cells need more fat and protein so they can grow and then divide. You are going to use the paper models of the monomers you cut during the digestion activity to model biosynthesis of fats and proteins. During this process, no new high-energy bonds will be broken or created, so the chemical energy is going to stay the same. All that we're going to do is change how molecules are combined. First, we're going to biosynthesize a protein. Take your 4 amino acids for the digestion activity. In order to link the amino acids together, you are going to have to remove the hydrogen and OH group that you taped onto the lonely carbons and nitrogens. After removing the H and the OH groups, this leaves sticky ends on those amino acids. Carbon and nitrogen atoms need more bonds in order to be happy. You can tape the amino acids together to create a protein. You also need to create water molecules using the H and OH components you just cut off. At this point, the video is going to pause to give you a chance to biosynthesize protein and answer a couple of questions. Here's an animation showing what you just did. The amino acids undergo a chemical change where the H and OH are removed, allowing the amino acids to be stuck together. Those H's and OH's then can stick together to make water. Since water is removed from the amino acids, this is called a dehydration reaction. Just like when you were low on water, we say you're dehydrated. When the amino acids lose water, they have also been dehydrated. Also notice that the number of carbon atoms and the high energy bonds stays the same in this process. All we are doing is rearranging the connections between these molecules. Now you are going to do the same thing for fat. Use your glycerol and fatty acids that you digested in the previous activity. Cut off the H's and OH's from each one. Put the glycerol and fatty acids back together and create water with the pieces you just removed. I'm going to pause the video to give you a chance to biosynthesize fat and answer a couple of questions. Here's an animation showing the dehydration synthesis of a fat molecule. Just like in the proteins, H's and OH's are removed from the monomers, leaving sticky ends that allow the monomers to stick back together to make a fat molecule. The H's and OH's join back together to form water. Notice how the number of atoms stays the same in this chemical change, and the amount of high energy bonds also stays the same. Going back to the cartoon version of our polymers and monomers, here's what's happening. The monomers move through the bloodstream and enter the cell through passageways on the cell membrane. Once inside the cell, they are biosynthesized to make larger polymers again. So what are these molecules used for once they get there? As you can see here, the fats are used in the membrane. Granted, they don't look exactly reformed to make the same polymer you started with. Instead of a glycerol and 3 fatty acids that we started with before digestion, the lipids in the membrane have 2 fatty acids instead. However, the monomers that are needed to build the cell membrane come from the fats you eat. If a cell is going to grow and get bigger, it's going to need a bigger membrane. For this to happen, it needs more fat. So what about sugar? You may have noticed that we haven't done much with the sugars so far. In animals, carbohydrates don't contribute much at all to the growth of an organism, but that doesn't mean that sugars aren't important. The diets of most animals consist of a lot of carbohydrates. The mealworms eat potatoes, the cow eats grass, I eat a lot of Cheez-Its, and I'm sure Christian Bale, with his Welsh roots, has eaten his fair share of fish and chips, which have quite a lot of starch. This means that lots of sugar ends up in our blood and travels to our cells. But animals don't make starch or cellulose like plants do. So what are animals doing with all that sugar? Turns out sugars are pretty critical in animals. They make fat and protein from sugar. They link glucose monomers together to make glycogen, which is a bit like a branched form of starch. This is how animals store glucose to be used later. Or animals can combine sugar with oxygen, which releases energy that an animal can use to function. We're going to go through each of these in a bit more detail. Let's start with how animals make fats and proteins out of glucose. Think back to the beginning of the digestion lesson. We saw that the grass a cow eats is composed primarily of water and carbohydrates and a little bit of protein. But the cow is made up of a lot of fat and protein. How is it that the cow is made up of protein and lipids if it doesn't eat much protein or any lipids? To answer that question, it helps to look again at the food molecules at the atomic molecular scale. Compare the atoms in glucose to those in fat. How do you think your body makes fat? If you said that the atoms from glucose get rearranged to make fat, you're right. In our bodies, if we eat too much sugar, that sugar is moved into fat cells where the atoms can be rearranged to make lipids. This is why when you eat too much sugar, you can get fat. How about proteins? Compare the atoms in glucose to those in proteins. What's different? So hopefully you noticed that proteins have nitrogen in them, an element that isn't in glucose. So where do cows get their nitrogen? To answer this question, we need to take a step back to the macroscopic scale. Remember when we zoomed into air? We saw that 80% of the atmosphere is nitrogen gas. Unfortunately, this form of nitrogen is not useful to animals. So where does the cow get the nitrogen it needs? Well, it comes from the plants. Plants can assimilate nitrogen into their bodies, then animals can eat those plants to get nitrogen. This nitrogen gets absorbed into the bloodstream and is transported to the cells where it can be used to make amino acids. There, it combines with the atoms in the glucose to make all of the non-essential amino acids. Those are the ones that you can make in your body. On to the second way animals use sugar, to make glycogen. This is the way animals store glucose to be used later. Glycogen looks a bit like starch, only it's a branched molecule. It is primarily made in the liver for storage. When other cells need glucose, the liver sends that glucose to where it is needed for the body. Even though the liver is where the glycogen is stored, this carbohydrate still only makes up about 5 to 6% of the mass of the liver. Muscles also store glycogen as they need lots of glucose. Even so, glycogen only makes up about 1 to 2% of muscle mass. So what does our body do with all that glucose? This is where the energy you need to function comes from. There are lots of high-energy bonds in glucose. This chemical energy can be released and transformed to other forms of energy when glucose is broken apart. This will be our next topic. To recap, we started by looking at the molecules in food. We know the polymers are carbohydrates like starch and cellulose, fats, and proteins, and that these polymers are made of smaller monomers. We looked at how the polymers were broken into their monomers during digestion, and how these molecules are rebuilt in cells in a process called biosynthesis. Finally, we looked at how sugars are used through a process called cellular respiration, which we will investigate in more detail in future activities. Now, answer a couple more questions about digestion and biosynthesis. Remember, if you feel shaky on any of this, go back and rewatch parts of the video. There are also additional resources for you to use in the Study Resources section of my website. When you feel confident about digestion and biosynthesis, fill in your summary chart for those two activities. Good luck!